In this section, we'll take a closer look at the external structures of prokaryotes. Beginning with the appendages, these are cell extensions. There are two types of appendages. Some are for motility and some are for attachment. Not all species have these appendages, but for those that do, it can provide motility that we'll see with flagella and axial filaments. Or, as we'll see with pili and fimbriae, they can be used for attachment and mating. Let's first take a deeper look into flagella. The flagella are composed of three portions: the filament, the hook, and the basal body. The filament's a helical structure. It's composed of proteins. It's only about 20 nanometers in diameter and about 15 to 20 microns in length. So naturally, it requires very high magnification to see these parts. It's unlikely that you'd be able to see them in your microscope. Now, flagella vary in number and arrangement. Some are in a polar arrangement and some are in a non-polar arrangement. In the polar arrangement, we see flagella are attached to either one or both ends of the cell. We can see that here in the monotrichous type, the lophotrichous type, and the amphitrichous type. Monotrichous means simply a single flagellum. Lophotrichous means there are small bunches or tufts of flagella, loph, emerging from either one or both ends. Amphitrichous is having a flagella at both poles, a single one. So you could have an amphitrichous, lophotrichous flagellar arrangement. Now, when the flagella are not polar, meaning they are all over the cell, it's called peritrichous. As you can see in this image, the flagella are dispersed randomly over the structure of the cell. Now, this whole flagella filament is inserted by a curved tubular hook, this region right here. And that tubular hook descends through the cell wall, through the outer cell membrane, and is embedded in the inner cell membrane. In this arrangement, it has the capacity to rotate in a 360-degree range of motion. And in most species, it can go counterclockwise as well as clockwise. We'll see how that works here shortly. Let's take a look at a quick animation to help us understand how the flagella attaches to the bacterial cell. All of these animations and more are available for you in Connect, under your library.
Most motile bacteria move by means of flagella. A flagellum is a slender structure about 15 to 20 microns long and about 20 nanometers wide. A typical rod-shaped bacterial cell is 2 to 3 microns in length. The bacterial flagellum is composed of three parts: the basal body, which is embedded in the cell membrane and wall, a short curved segment called the hook, and the portion that extends out from the cell called the filament. The basal body consists of rings that correspond to the layers of the cell envelope and therefore differ in gram-negative and gram-positive bacteria. In most gram-negative bacteria, there are 4 rings connected to a central rod. Gram-positive bacteria have only 2 basal body rings, one connected to the cell membrane and the other to the cell wall.
So what is it that causes bacteria to move? Flagellated bacteria can detect and move in response to chemical stimuli. This is called chemotaxis. It can either be positive or negative. In positive chemotaxis, the movement is in the direction of a favorable chemical stimulus. Usually it's a nutrient, something they'd like. Negative chemotaxis is movement away from a repellent, usually a harmful compound. Their mechanism for detecting chemicals just so happens to be tightly linked to flagellar function. Now, we're just beginning to understand the molecular details of this, so if you want to watch the animation about that in Connect, you're welcome to, but I won't hold you responsible for the specifics. Some photosynthetic bacteria will exhibit phototaxis. That is, they move in response to light. Naturally, they'd need to move towards it if they need to perform more photosynthesis. Let's take a closer look at how they actually move. They move by runs and tumbles. I can show you what that means here in this figure. When we look in figure A, a counterclockwise spin of the flagella will propel the bacteria forwards. Every so often, that spin changes direction and the cell stops and tumbles or changes its course. You can see these patterns exhibited in the figure below. We'll talk about those shortly. In peritrichous forms of bacteria, the flagella sweep towards one end and then they rotate together. If they're moving in the counterclockwise direction, they move forward, and if they move in the clockwise direction, here they tumble again. In the presence of attractant molecules, bacteria exhibit more tumbles. So it's thought that these attractant molecules may inhibit the tumbles and permit the progress of the bacteria towards the direction of the positive source. In the case of repellent molecules, they cause numerous tumbles. And it's thought that these tumbles allow the bacterium to redirect itself. You can see this exhibited here in the picture below. Each time we see a red dot, it's a tumble. The green stripes are runs. So we alternate tumble, run, tumble. If there's no attractant or repellent, they move in sort of a random format like this. However, when there is an attractant, they still run and tumble, but it is in a general direction. Let's watch a quick animation that displays this behavior of chemotaxis in Escherichia coli.
When Escherichia coli moves in a medium that lacks a concentration gradient, the cell travels, stops, or tumbles, and then continues moving in a new random direction. When the flagella rotate counterclockwise, the flagella form a tight bundle and propel the cell forward in a run. After a brief period, the direction of rotation is reversed, causing a tumble. As cells move up a chemical gradient, the runs are longer than when they travel down the gradient. The overall result is random movement in the absence of a chemical gradient and movement toward a chemical when a gradient exists.
You can review this animation anytime simply by going out into Connect and looking at the videos under the library. The rest of the animation shows how this movement is linked to chemical sensation, chemical receptors. The flagella of spirochetes present a unique situation. Spirochetes have this unusual wriggly form of movement. And that's because they have these periplasmic flagella, or axial filaments. They're located in the periplasm, so in the area between the cell wall and the cell membrane, right here in this space. They're represented by white dots in this case. There are bundles of many flagella throughout the periplasm, and they contract and cause the bacterium to form this kind of corkscrew movement. This motion can be seen in the spirochetes of syphilis. Let's take a quick look at this animation to see how the spirochete movement works.
Spirochetes are motile by means of bundles of flagella, called axial fibrils, that run along the cell within the periplasmic space. The number of fibrils varies. These axial fibrils extend from both ends of the cell within a flexible outer sheath. Spirochetes have a screw-like motion which allows them to bore into mucoid layers and soft agar media.
Now let's take a look at the appendages that are used for attachment. Some of these appendages are also used for mating. We'll take a look at fimbria and pili. Fimbria are used for attachment and sometimes for motility. They allow bacteria to attach to each other and to other surfaces. In fact, they may be responsible for the mutual clinging of cells that cause biofilms and other thick aggregates of cells on the surface of liquids. All it takes is a few to begin, and they may be responsible for creating the foundation for complex communities to develop. The sex pilus, or pili in the plural form, are used for attachment in order to accomplish genetic exchange. So it's not about movement here. The pilus is a rigid tubular structure that's made of protein. So far, they've only been found in gram-negative bacteria. Gram-positive bacteria have another mechanism for exchange of genetic material. How about a quick concept check? What type of flagellar arrangement has appendages at both poles of a rod-shaped cell? Monotrichous, amphitrichous, lophotrichous, or peritrichous? Mono means one, so that wouldn't cover both ends. Amphi means two, And amphi literally means both, so that covers both ends. Peritrichous means all over, so we'll get rid of that. And lophotrichous means in clumps. And they could be on one or both ends, so that one's kind of a maybe, but the best solution is certainly B. As we move through more structures on the exterior of the cell, we'll consider the glycocalyx. The glycocalyx develops as a coating of repeating polysaccharide units, protein, or both polysaccharide and protein. Its function is primarily to protect the cell. Sometimes it can help the cell adhere to the environment around it. And it differs between bacteria in thickness, organization, as well as chemical composition. There are two basic forms. You can have a slime layer that you see here in the green-coated bacteria. This slime layer is what would help formation of a biofilm. And the slime layer is more of a loose shield, and it protects the bacteria from loss of water and nutrients primarily. Some bacteria form a capsule out of the glycocalyx. You can see that over here. The capsule is bound much more tightly to the cell and is much more dense and thick. In the prepared slide here, you can see a negative staining that reveals large, well-developed capsules around these bacteria. In the petri dish below, you'll also see that there are colonies produced in the petri dish. Those on the left are non-encapsulated, and those on the right are encapsulated, so they form more of a sticky colony in the agarose gel. Let's look in more detail at the functions of this glycocalyx. Sometimes we'll see the capsule formation. It's formed by many more pathogenic bacteria. Those are the ones that are trying to invade our systems. This capsulation protects the bacteria against our phagocytes. You'll recall our phagocytes are the white blood cells that engulf and destroy invading cells in our body. The slime layer is important again in formation of the biofilms. As I just said, plaque on our teeth comes from these surface slimes that are produced by certain streptococcus bacteria in our oral cavities. This slime prevents them from being dislodged and also provides a really nice niche for the growth of other microbes. Some of these slime layer covered microbes are so sticky that they allow colonization of plastic catheters, like you'll see over in this image here on the right. You can see the gray area is the catheter surface, and then you can see the glycocalyx slime accumulating over here around various colonies or clusters of cells. Not only do they form on catheters, we also can sometimes see them on IUDs, intrauterine devices that are used for birth control, and often on pacemakers. So as you can see, it's really important to employ sterile technique to avoid these kinds of bacterial growths.